Good day everyone. Today we're going to present a report on the denaturation and renaturation of salivary amylase. In 1972, Christian Alvinson wanted to show that the information for protein folding resided entirely within the amino acid sequence of the protein. He chose bovine pancreatic ribonuclease A as his model for folding, but he couldn't completely denature the protein unless he treated it with a denaturant urea plus beta-mercaptoethanol to break the disulfide bridges. Then, upon the removal of mercaptoethanol but not urea, Anfinsen discovered that the enzyme spontaneously refolded into a scrambled structure with no catalytic activity. This is attributed to the formation of random disulfide bridges between the 8 cysteine residues present in the protein. It also shows that the correct three-dimensional conformation must be achieved fairly rapidly when urea is removed, since most of the proteins under those conditions becomes active. But upon treatment with trace amounts of mercaptoethanol to break once again those random disulfide bonds and removal of urea from the solution, ribonuclease A refolded spontaneously and regained its biological activity. This demonstrated that refolding could take place in vitro. However, recovery is not 100%. Mistakes are made in vitro and presumably in vitro as well. Furthermore, the correct refolding must take place in an environment with the correct and specific pH and temperature. Nonetheless, his discovery led to the conclusion that the information required for a polypeptide chain to fold into its active shape must reside in the amino acid sequence rather than be imparted from anything else during protein synthesis, such as DNA, RNA, ribosome, and enzymes. This won him the Nobel Prize for Chemistry in 1972. Then in 1973, Anfinsen followed the proposal for a thermodynamic hypothesis to account for the protein folding. He stated that the three-dimensional structure of a native protein in its normal physiological milieu, defined by properties such as the solvent's use, its pH, ionic strength, presence of other components such as metal ions or prosthetic groups, and temperature, is the one in which the Gibbs free energy of the whole system is lowest. That is, that the native conformation is determined by the totality of interatomic interactions and hence by the amino acid sequence in a given environment. This also clearly established that genes determine the amino acid sequence, and the sequence in turn determines the active shape and structure of the enzyme, and this ultimately determines their biological functions. To adopt Anfinsen's study, this experiment will demonstrate the denaturation and renaturation of salivary amylase in varying pH and temperature. In the human body, alpha amylase is part of the digestion and the breakdown of carbohydrates in the diet. It is a hydrolase that acts on alpha-1 for glycosidic bonds and catalyzes the hydrolysis of alpha-linked polysaccharides like starch into alpha-anomeric products. Once the enzyme reaches the stomach, it becomes inactivated due to the acidic pH. Further breakdown of starch occurs by secretion of a secondary form of the enzyme by the pancreas. Pancreatic juice enters the duodenum and pancreatic alpha amylase further cleaves starch to either yield maltothiose and maltose from amylose or maltose, glucose, and limit dextrin from amylopectin. Hence, for this experiment, the objectives are to demonstrate and observe protein denaturation and renaturation of salivary amylase, to determine how varying temperature and pH affect protein denaturation and renaturation, and to determine the effects of temperature and pH in the catalytic activity of salivary amylase through spectrophotometry.
The methodology is split on control confirmation and setup, effect of temperature, and effect of pH. In order to determine the change in structural stability, we will monitor its biological activity through starch hydrolysis using iodine potassium iodide solution as indicator. We must set up the solution and the experiment apparatus. We start on preparing a human saliva solution at a ratio of 1 to 10 at a final volume of 50 milliliters using 0.8% NaCl solution. We filter the solution to remove the particles. We then set up the experiment setup following the configuration shown in the slide. The thermometer is immersed in the enzyme solution, whereas the beaker holding the enzyme solution is placed in a water bath. The water bath serves to set the temperature of the environment. Magnetic stirrers is placed on both the water bath and the enzyme solution. We will first check if the enzyme solution is working. At the initial temperature, 500 microliter aliquot of the solution is taken and placed into a positive control tube. The solution is left to incubate for 30 seconds before being terminated by 100 microliters of concentrated sulfuric acid. The negative control is made by placing the sulfuric acid first before adding a 500 microliter enzyme solution. A 10 microliter indicator solution is added to both positive and negative control to see for differences. The experiment is resumed if there is a difference between the coloration of the solutions. After confirmation of the positive and negative control, we copy the positive control part of the control confirmation. We take a 500 microliter aliquot of the enzyme solution and add it to a start solution and leave it to incubate for 30 seconds. It is then terminated with 100 microliters of concentrated sulfuric acid. We will then repeat the process at every 10-degree increment starting at room temperature leading up to 60 degrees Celsius. After reaching the maximum temperature, we transfer it to a separate plate and repeat the experiment but with the cooling of the solution until room temperature. For each solution obtained, 10 microliters of the indicator solution is added and diluted to 10 milliliters. It is left to stand for 20 minutes before taking the adsorbance reading at 620 nm. We repeat the first three steps: the preparation of enzyme solution, preparation of experiment setup, and the confirmation of positive and negative control. The pH experiment is similar to the temperature experiment, but the solutions obtained came from adding 0.1 molars of sodium hydroxide from initial pH to pH of 12 and then titrating it back to initial pH with 0.1 molars of hydrochloric acid. Now, let's plot the data obtained from the effect of temperature and effect of pH datasets. The temperature plot have shown a linear trend in both the warming up and cooling down part of the experiment. The initial temperature is 25 degrees Celsius corresponding to an absorbance of 1.21 on the warming up and 1.22 in the cooling down. Increasing the temperature at both warming and cooling setup has an increase in absorbance, indicating that there is an increasing amount of denatured protein as more starch is left undigested. The warming up curve started with 1.21 at 25 degrees Celsius and ending up on 1.75 on 55 degrees Celsius, whereas the cooling down curve started with 1.38 at 45 degrees Celsius and ending up at 1.22 at 25 degrees Celsius. The optimal temperature in this temperature set is determined to be 25 degrees Celsius at both warming and cooling. The absorbance plot at varying pH have shown that a similar trend is followed, wherein an increase in pH resulted to an increase in denatured proteins, and this trend is consistent with both the increase in pH via NaOH and decrease in pH via HCl. However, the difference in values between the increase and decreased pH are significantly higher upon compared to similar values. At pH 11, the increase in pH data point indicating an absorbance of 0.63, but the decrease in pH curve has 0.8, indicating an increase of the denatured protein upon reverting back to the solution's pH. The optimal pH is determined to be 8.6 for the increasing pH and 7.1 for the decreasing pH.
Now for the discussion part of the experiment. For the triiodine complex, iodine is not readily soluble in water and therefore is dissolved with potassium iodine, which eventually dissociates to yield triiodide ions that can enter the helical structure of amylose and starch. The formed complex between amylose and iodine experiences a charge transfer that changes the gap between the orbitals of their atoms, resulting in changes in color and absorption wavelengths. The different stages of hydrolysis of starch by salivary amylase is indicated by different colors. For the iodine coamylose complex, the blue-black absorbance has a maximum absorbance of 620 nanometers. Spectrophotometry can therefore be used for analyzing samples tested with the iodine reagent to determine starch or glucose or maltose concentration for the purpose of monitoring salivary amylase activity. At optimal temperature, pH, the absorbance for the complex will be at its minima, where amylase activity is highest. This implies that more of the starch is being hydrolyzed by salivary amylase into glucose or maltose, and thus the concentration of starch decreases, and consequently, less complex that absorbs energy at this wavelength would be formed.
We have seen in both pH and temperature plots that the function and thus the structure is altered upon increasing or decreasing the pH and temperature. But how exactly? The change is centered around the alteration of H bonding and electrostatic interactions. The change in pH induces charges between the amino, carboxylic, or the side chains, resulting to the changes in electrostatic interactions. The change in temperature causes changes in the vibration of the domains, leading to an alteration to the structure. Renaturation is conducted by reverting to the environmental conditions back to the conditions of the native state.
The thermodynamic hypothesis of Anfinsen stipulates that the three-dimensional structure of a native protein is governed by its physiological conditions. At optimal conditions, it gives free energy of the whole system's lowest. However, the condition associated with the thermodynamically lowest energy state changes upon environmental perturbations, which ultimately leads to protein denaturation or the disorganization of native protein structure. During denaturation, the total energy that arises from the enthalpically favorable interactions of polar side chains and water molecules and unfavorable ordering of water molecules surrounding the protein is displaced. For instance, increasing the temperature provides enough energy for the protein to escape its energetic minima and transition into another conformation. This happens while keeping the peptide bond intact, hence conserving the amino acid sequence. But the consequent process of denaturation results in the loss of secondary, tertiary, and quaternary structures, which ultimately results in the loss of its biological function. Now, the optimal activity of proteins depends on conditions such as pH and temperature. But aside from these two, other physical and chemical agents of denaturation include X-ray, UV, heavy metallic ions such as lead and zinc ions, and highly electronegative ions like cyanide, organic chemicals, and chemotropic compounds like urea. For instance, metallic ions can form ionic bonds with the carboxyl group, while highly electronegative ions can form ionic bonds with protonated residues. This can disrupt the ionic bonds within the protein. Metallic ion salts can also disrupt disulfide bonds due to their high affinity and attraction for sulfur. On the other hand, organic chemicals have favorable interactions with hydrophobic residues found in the inner core of the protein, allowing these residues to come to the surface and consequently unfold the protein. They can also disrupt hydrogen bonding. Protein denaturation often results in an observable increase in protein viscosity due to their aggregation and increase in ionizable groups. To facilitate refolding into its native state, the optimal environment for the protein must be met. This also includes the following factors or conditions. First, optimal folding conditions, which differ for each protein, and restoration of physical and chemical environment, such as the removal of denaturing agents. Also, the amino acids should be undamaged, unchanged, and have not formed new bonds. It would be also easy to renaturate a protein if disulfide bonds are not essential for its enzymatic activity. And lastly, it is preferable that the protein does not require or possess cofactors in its structure. Often in practice, temperature is first gradually reduced during protein refolding to safely restore the native conditions of the protein. If a sudden temperature change is performed, the protein may be trapped in a local minimum rather than reaching its native state, with insufficient energy to escape the local minimum. Then, chemical denaturants are removed through methods such as dialysis. These include chemotropic compounds and reducing agents like mercaptoethanol. Some of these techniques were actually employed in Anfinsen's experiment. Moreover, transferring the protein in a solution with a compatible or adjusted pH to native conditions and osmolarity may also be performed. A biological pH range of 6.5 to 7.5 is commonly used for aqueous proteins, while membrane-bound proteins, which are relatively harder to renaturate, need the compartmental pH of the membrane they came from. Lastly, other renaturation techniques can be performed such as dilution of denaturing agents, chromatographic techniques, and use of chaperones in some cases to assist the refolding of proteins. Successful denaturation procedures are often indirectly determined.
The plots have shown the same trend on both increasing and decreasing values, but the data points are not entirely the same. This observation indicates that not all 100% of the denatured proteins are renatured back to the native state. The differences in data in the temperature plot range from 0.01 to 0.06, whereas the differences in the pH plot could reach as large as 0.53. The decrease in proteins at the native state can be attributed to the incorrect non-covalent interactions. As the number of renaturation and denaturation cycles increase, the number of incorrect interactions increase. Additionally, Some proteins have features that do not allow complete renaturation, as it requires additional factors such as the presence of important disulfide bonds and cofactors, which were mentioned early on in the presentation. Learning denaturation and renaturation of proteins, we might ask what are the physiological and biological advantages of protein renaturation? Renaturation decreases the consequences of environmental changes by minimizing the loss of bioactivity. Renaturation in biological systems are known to be conducted by chaperones and binding proteins. An example of renaturation in place is a study conducted by Schumacher et al., where chaperones renatured thermally denatured luciferase, thereby providing evidence of systems present on minimizing damage due to thermal changes. Additionally, renaturation prevents the accumulation of misfolded and denatured proteins in biological systems, decreasing the possibility of disorders. An example of a disorder that is formed when renaturation and refolding mechanisms are inadequate is Alzheimer's disease. Among the different array of proteins involved in Alzheimer's disease, beta-amyloid is one of the most studied. A study conducted by Balgun et al. Have used Lepidium sativum extract for treatment of aluminum chloride-induced AD and has shown beta-amyloid management and protein renaturation. Studying such phenomena provides further insights to the behavior of proteins under strenuous conditions. The knowledge obtained from denaturation-renaturation studies have been used in medicine and further protein research. This includes increasing protein yield from recombinant organisms with modified extraction processes, production of solutions that induce protein renaturation such as ionic liquids and non-detergents for phobetanes, and the development of techniques for studying enzyme catalysis.
Thank you for listening, and here's the list of our references.